Hi, this is David Sankoff, and I'd like to talk about combining synteny and similarity-based inference on the polyploidization-fractionation cycle. In plant comparative genomics, a common type of data is a gene pair similarity distribution. The peaks in this distribution represent ancient polyploidization events. So here we have one triplication event, also called hexaploidization, followed by a duplication event called tetraploidization. We resume this in the following type of diagram, where the triplication's at T1, the duplication's at T2, and the observations of the genes surviving fractionation are at the present time, and these are all the trajectories. This is summarized in the probability measure P, where R is the ploidy vector, 2, 3, 4, whatever. Mi is the number of genes in generation I. Each of these genes gives rise to R copies, and A counts the number of genes for each of these copies that survive. U is the retention rate, or the multinomial probabilities. We also have the partial trajectories, the measure on the partial trajectories between the H and K generations. However, this only generates the number of genes in each generation. We need, for these data, the number of pairs of genes. To get that, we can multiply the expectations from three independent events, one from M1, from the time 1 to time Ti, count the number of pairs, and then for each of these pairs, count the number between Ti plus 1 and the present day, and we can predict the number of pairs. Unfortunately, there are too many parameters for this type of data. There's too many parameters to explain this type of data using our branching process. For example, in this case, we have two equations. The measure reduces to these two equations in this case, and there are three parameters. U is the probability of retaining two genes instead of one. U prime is the probability of retaining three, and V is the probability of retaining two after this second event. To get around this, we look at the synteny block that gave the original data. These synteny blocks are subject to fractionation, and you can see the empty circles represent genes that are lost through fractionations, and each one of them has a partner which is not lost, and we count these singletons and we can predict them using the same measure. So now, instead of two equations for three parameters, we have four equations for three parameters, and this gives us the luxury of adding in another parameter that we can estimate the number of genes in the original population before the first event. Unfortunately, there's another problem with the data, and that is the earlier events give rise to synteny blocks which we cannot observe very well. We can observe them, but they've lost a lot of genes. Some of them have disappeared because they're not long enough for the software, and the software won't accept synteny blocks that are shorter than some default, and there's a general downward bias to estimating the MI for the early events, and an upward bias for estimating the parameters, and this becomes very severe. We can get around this by just multiplying each MI by a constant, which we can infer if we have enough equations. So this becomes C, the crumble constant is added in two of the equations. And this happens in general whenever we have more parameters than 2 per more retention rates than 2. If Ri is bigger than 2, we have extra parameters. However, we can get around this thing by assuming that the number of genes that are lost follow a binomial distribution based on a loss probability of p, and with that we can find that in this case that the u prime, the probability of retaining 3 genes, is somewhere between u squared and u cubed, as long as u is not too big, between 0 and 0.4. Now I'd like to talk about 5 plants that we've analyzed, among many others, which have different histories. So the durian has had a triplication and followed by a triplication. The first triplication on the left is the gamma distribution at the origin of the core U dicots. And I'll just say that if you look at the number of genes estimated for the original population, you get over 16,000. For poplar, which is a triplication followed by a duplication, we get 17,000 using that model. For linen, which also has the same early gamma triplication as the other two, we get 16,000 for the estimate. Black pepper, which I just showed because it's not a core U dicot, it has a duplication followed by a duplication, and we get a different number of genes at the origin, of course, different part of the plant phylogeny. And finally, salvia, which has 3 events. We don't know what the middle one is, but whatever one we use, we get around 14,000, not far off the other ones. Thank you.